Okay, good afternoon. Thanks for the opportunity to talk. So my name is OJ Tucker out of Goddard Space Flight Center. And so I want to be talking, similar to the other talks, I'm going to be talking about how hydrogen implantation leads to that 3-micron absorption feature attributed to OH or water. And so in this simple study, we're looking at how when you go into magnetotell and the solar wind is blocked, how that decrease in proton flux affects the surface concentration of hydroxyl and also hydrogen content in the exosphere. And I'd like to thank my co-authors, Bill Farrell and Andrew Poppy. Okay, let's see. Oh, okay, yes. Shouldn't be that hard to figure out, huh? So just to start off with, we're trying to understand this lunar hydrogen cycle, right? And so it's this really complex process where you have various different sources, solar wind, providing protons for implantation, meteoritic sources. You also might have ancient sources at the poles that can all contribute various forms of hydrogen to the lunar surface and also in the exosphere. So implantation can implant hydrogen and it can combine with other atoms and molecules in the surface, forming various molecules like methane or hydroxyl. So, as any good scientists, we want to try to break this problem down to understand it. And so what we're focused on is how proton implantation forms hydroxyl in the surface, and also how hydrogen diffusion out of the surface sources the H2 exosphere. And so these have been observed in various different forms. So we have over 5 years of ion data of proton flexes to the lunar surface from Artemis. So in the sheath region, the proton flux is about a factor of 2 larger than the nominal solar wind flux outside of the magneto sheath. At the magnetotell, the proton flux is actually the average proton flux is lower by about an order of magnitude. We also have information on the H2 exosphere in the tail by Chase, about 500 to 800 cc. We have some information by LAMP on H2 in the exosphere. LACE performed some of the initial measurements, provided upper limits. And also of this absorption feature, we have the M-cube data, which we'll be talking about. So it's also some M-cube data in and out of the tail that show very small changes in the tail compared to out of the tail, a decrease in the absorption in the tail compared to out of the tail. So we think understanding whether it's OH or H2O in this implantation process, we need to connect a variety of observations together, and we try to do that through modeling. So this is the general idea. Protons come in and implant. They become neutral. So those protons that are implanted also cause damage. They create defects in the regolith, space weathering. Hydrogen diffuses out. One of those defects can be dangling bonds. The hydrogen can become attached to those dangling bonds and form hydroxyl. So this theory was put forth by Star Keenan, who did a lot of work on this. And so we can represent that with like a diffusive lifetime, and we can get implantation depth from experiments. We can get activation energy of the diffusive path. So you can imagine that diffusive path is changing with every implantation. And we can also get diffusion coefficients from experiment. Bill Farrell did a lot of work showing how you can model this process by using a distribution of activation energies, because you can imagine the surface is constantly changing. Those defect sites are constantly changing. And so, based on the amount of defects in the surface or the type of defect, you can have hydrogen remaining for, the history of the moon or just for a fraction amount of time. So the simulation is just very simple, taking advantage of the synchronous orbit of the moon. We can define a local time for when the moon is in the sheath, and we can increase the flux by a factor of 2. In the tail, we can decrease it by an order of magnitude for 5 days. So the source is just the proton flux, just basic loss processes, thermal escape, photo destruction. And the Monte Carlo part comes into place with a distribution of implantation depths based on the distribution of incident ion energies, which have been obtained from Artemis data. And then the distribution of activation energies, which has been shown to be represented in experiment very well using a Gaussian distribution. And when they leave the surface, just the Maxwell-Boltzmann velocity distribution, tracking the H2 that comes out of the surface. And so we did some previous work on this before, not looking at how it changes in the tail, but just looking at how hydroxylation changes over the day. And we found that we could reproduce the M³ surface concentrations, so that increase at high latitudes up to about 700 parts per million. And also the diurnal variation, we could reproduce that fairly well using just using experimental parameters for diffusion coefficient, activation energy. So this characterizes the Gaussian distribution of activation energies here. And so what we found is if the activation energy was just a little bit larger than what experiments have derived, you would have too much retention of hydrogen, and the surface concentration would be much larger than M³. And if it was too low, you wouldn't retain enough of hydrogen in the surface. And this is just showing the previous result compared to the data here. So results from our new study. This is showing the surface concentration of hydroxyl. So red is showing hydroxyl content up to 800 parts per million, from 200 parts per million up to 800 parts per million. Blue is below 200 parts per million. This is a surface map shown here. So the little sun point just shows the subsolar point. And so if you take a snapshot during full moon, you get a surface distribution like this. And if you take the densities along the equator, so at noon you get, surface concentrations about 5 parts per million. On the night side, it's about 100 parts per million, and then at dawn, so it decreases over the day. All right, now if you took a snapshot at new moon, you get two minimums here. So you get one related to the subsolar point and one related to the decrease in flux as the moon is going through the magnetotell. And so if you were to compare the hydroxyl content at the subsolar point during new moon and full moon, you wouldn't see very much difference. But if you were to look at the night side on a part of the surface that's constantly experiencing low flux as you go through the magnetotell, you could possibly see a difference in the surface content due to the decreased proton flux. Okay, and so this is just a comparison. So I grabbed this from Schweili's LPSC result a couple of years ago, and it's just showing just a slight difference between the absorption feature in the magnetotell compared to before the magnetotell and after the magnetotell at about mid-latitudes. And just doing a little comparison here to our model result for full moon shown by the solid red line when it's in the magnetotell, and new moon when it's outside of the magnetotell, showing a relative difference here, just a relative difference. So that's looking at looking along subsolar latitudes at local noon. All right, so moving on. So what about the exosphere? So looking at the molecular hydrogen that's diffusing out into the exosphere, this is showing the distribution during full moon here from day to night shown here. So if you were to take it about the equator, just a profile about the equator, you get this line shown here. And so this is the surface concentration ranging from about 400 cc at noon, and then on the night side, because it's accumulating due to the lower temperatures, you get about 1600 cc. And it's consistent with some analyses of lamp in the tail from LPSC abstracts. But then if you look during new moon, when it's not shielded, you get a much larger hydrogen content, up to 4,500 at night and 2,000 at noon during the day. And so we also did some comparison to chase in the magnetotell. So if you were to look at the densities in the magnetotell, in the magnetotell during full moon, when I believe chase was operable, you can see we also get consistent densities for that as well due to the decreased source. And so these plots here are showing, so if you were to imagine being on a point on the surface and measuring the atmospheric concentration sort of using a mass spectrometer over the time of day, if you're at the sub-Earth point, you will get a distribution like this as night comes. Then during the day, because you're shielded by the full moon, you get this dip of about an order of magnitude due to the decrease in flux. And this is at the anti-Earth point shown here. So really what concentration you get really depends on time of day and also the location that you're at on the moon when you're making your observations. So we can connect these things to understand the physics that's driving them. And yeah, so just showing here that the independent observations of H2 between chase, between lamp, they seem relatively consistent. You would expect that, so this is a plot that I took here from Cook's LPSC abstract that compares how helium changed in the tail compared to hydrogen. But helium is much larger, helium, because it weighs larger, you get a larger accumulation in the exosphere, so you can see changes more easily with helium compared to hydrogen. So that might have been like within the error bar limits that I shown in the earlier plots. But hydrogen could be consistent with all of these different observations depending upon location and time of day. So with light species that are lost relatively quickly, you would expect to see a change. If it's a solar wind source, you'll expect to see a change while it's in the magnetotell for less than 5 days. But for heavier species like neon and argon, you probably wouldn't see such changes because they stick around for a long time. And with that, I'll just leave up the conclusion. Thank you. Awesome.
Thank you. We have got a couple of minutes for questions. Dana?
OJ, can you comment on the percentage of hydroxyls that are produced that are then converted into H2 in the exosphere? Is there a balance or an imbalance in that?
Right. At low latitudes, there's definitely a dynamic balance between the amount of hydroxyl that changes throughout the lunar day compared to the amount of H2 that's released into the exosphere. Yeah, so we have like a dynamic steady state at low latitudes. At high latitudes, you have a little bit of accumulation over time because of the low temperature. The diffusive lifetimes are much longer, and so you get buildup, but that would be countered by space weathering and other processes.
So you had a local time plot of the concentration and it was high on the night and then it dropped down, like it was one of your earlier plots. It dropped down on the day side, but it like started to drop before dawn. And I was wondering what causes the concentration to change before dawn? Is it back a little further? I think it was did you have like an OH one?
Oh, OH.
Yeah.
Oh, okay. Let's see here. Oh, I'm sorry. I don't know how to use this thing.
It was like really early on. There. Yeah, so it's starting to drop off, like, pretty well before dawn. Do you know why that's happening?
Yeah, it could be that I just didn't line it up properly, but yeah, I have to think about it more. Yeah.
All right.
Okay, thank you very much.